Hello everyone and welcome to another class of Physiology for Engineers with Professor Gonzalez-Fernandez at Lehigh University. Today we are going to continue looking at the kidneys and kidney function, and we are going to focus in the process of renal reabsorption and filtration, which is a process by which our urine is going to be formed and also our body is going to reabsorb all the nutrients and water that we need for our day-to-day activities. So this happens in the nephron. I made a small schematic of the nephron here. I know it's very complicated, but this is the best that I could do to summarize all the processes that we're going to look at today. In the previous lecture, we have a look here at the renal corpuscle, specifically at the glomerular filtration process that happens between the glomerulus structure, which has the Bowman capsule, and also the different blood vessels that are going to bring the blood to be filtrated. But then the nephron also has all these tubular structures that are going to participate in this process of reabsorption and filtration. So just to have a look at the different flow of the fluids here, the unfiltrated blood comes through here, goes to the glomerulus, it gets filtrated, and then the filtrated blood comes through here in this direction, back here, and back to our body, outside of the kidney. And then in the other direction, we have the tubular fluid. So the tubular fluid is everything that we have reabsorbed here in the glomerulus, or that we have filtrated at the glomerulus. So the tubular fluid is going to go from the glomerulus through the different parts of the tubules and the tubes, back here, and then it's going to go to the collecting duct to be disposed as urine. So there are different sections in the tube structure that are going to participate of this reabsorption and filtration process. So let's dissect all these zones and areas of the tubule by one. So first of all, we have this first section here. So the first section here, which is closer to the glomerulus, and that's why it's called the proximal. So this section that is closer to the glomerulus is called the proximal convoluted tube. And this section is really important because here we are going to reabsorb most of the water, the glucose, and the ions that we need to reabsorb in our blood. So in this zone here, we are going to reabsorb water, we are going to reabsorb glucose, which is very important, right, because we need energy. We are going to reabsorb sodium and chloride ions. We are going to reabsorb potassium. We are going to reabsorb phosphate. We are also going to reabsorb calcium, and we are also going to reabsorb amino acids. So most of these components are reabsorbed here at the proximal part of the tube. And this is due to two different processes. One of them we have already seen when we were looking here at the glomerular filtration. So these are the starting forces. Which is the hydrostatic pressure, and also they call it osmotic pressure. In this case, we are going to have different ones. The most important ones are going to be the ones in the interstitial space, which is the space that separates the capillary from the tube. And also it's important the hydrostatic pressure and the osmotic pressure in the capillary, in the blood capillary here, that is going to absorb and reabsorb all these ions and nutrients. The second mechanism that we have to take into account are the different tubular factors. So the tubular factors are characteristic of this part of the tube. The first one that we have to take into account are the glucose co-transporters. So here in the membrane of the proximal tube, we have these transporter proteins. So these transporter proteins, they are going to transport glucose. So the glucose is going to leave here at the tubular fluid and is going to be reabsorbed into the bloodstream. And at the same time that the glucose is leaving the proximal tubule, is also ions, for example, the sodium and the chloride, they are also going to leave with the glucose back into the bloodstream. So this is the first one, which is the glucose cotransporters, and the second one are also the hormones. So these hormones can either activate this reabsorption process. So these are going to be, for example, angiotensin 2 and aldosterone that we're going to look a bit more in depth in a sec. And also we can inhibit this process. So for example, hormones that are going to inhibit this process are dopamine, and also we have the atrial natriuretic peptides, which are going to avoid this process of glucose, sodium, and water reabsorption here at the proximal tube. Okay, so let's go back to the second different part of the nephron, which is important for the reabsorption process, which is the loop of Henle. So this second structure that we have to look at to understand what is happening here and the reabsorption and filtration process is the loop of Henle, which is like this structure here that we can see in the schematic. Okay, so in this loop of Henle, we have two different limbs. So we have this limb here in which the tubular fluid is going to go down, right? So this is the descending limb. And a very important factor of this descending limb is that it's only permeable to water. So this means that only water can leave the descending limb back into the bloodstream and be reabsorbed in the bloodstream. So this is the first structure. The second structure that is important here at the loop of Henle is this other limb that is going to take the tubular fluid up back into the collecting duct. And this is called the ascending limb. Okay, so this is very important because this is not permeable to water. So that means that water here cannot leave the loop of Henle. Therefore, the only thing that can leave here are the different ions, for example, the sodium and the chloride. Okay, so this descending limb and ascending limb together with the blood capillary here form this system that is called the countercurrent system or countercurrent multiplication system. So this is called countercurrent because we have two different fluids that are going to have two opposite currents. First of all, we have the blood here. So the blood that is filtrated from the glomerulus comes here, back here, back here, back here, and comes in this direction. So it comes here, 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 back to our body. But then we also have the tubular fluid that is running in the completely opposite direction. So instead of going this way, the tubular fluid is going this way, so opposed to the direction of the direction of the flow of the blood, as we can see here. And this is called the countercurrent multiplicator system. So this happens, and the filtration here happens thanks to a concentration gradient. Okay. So when the tubular fluid comes here to the descending loop of Henle, because the tubular fluid is going down, here remember that this descending part is only permeable to water, so the water is going to leave. Therefore, the osmolarity here, because the water leaves, here is going to, the tubular fluid is going to become more concentrated here. So it goes from 300 milliosmol to 600 here, and finally here to 1,200 milliosmol. Okay, so it goes to this direction because here water is leaving. Therefore, it's going to reducing the reducing the amount of water and increasing the concentration of solutes here in the tubular fluid. But as water leaves and goes into the capillaries, here our capillary becomes less concentrated because there is more water. Okay, so here it will be very concentrated, then it comes back up, and because of this transfer of water, it becomes less and less concentrated until it leaves the nephron. But what is happening here to the other side? So in the other side is happening the contrary. So remember that here the ascending limb, so here the ascending limb by which the tubular fluid is going to come to the collecting duct, in this one is non-permeable to water, so the water is not going to leave. But what is going to happen is that then the ions are going to leave. Okay. So the chloride and the sodium, they are going to leave the fluid. They are going to go into the blood, and because the water is not leaving. The water is not leaving, but the ions are coming out, is going to become less concentrated. So as it goes up and the ions are leaving back to the capillary, the tubular fluid becomes less concentrated. So it goes back to this 600 milliosmol, and then 300, and finally here to the 100 milliosmol. But here, as the sodium and the chloride leave the tubule, the ascending limb, and go into the blood, they are going to make the blood here, they are going to make it more concentrated. So first we start like at around 300. As more sodium and chloride goes into the blood, it's going to be more concentrated, and it goes back to this 1,200. Okay, so this is the contact current system. Just remember that we have the ascending limb, descending limb. They have different permeabilities, and these are going to create this concentration gradient, which is going to lead to the reabsorption and the filtration of water and solutes. Okay, so let's go for the third part that we're going to look at today.